So we are on module 9A. Notice it's called Prokaryotes and Viruses.
That's my shoe again.
Okay. First, we're going to start off with something very basic in biology, and that is taxonomy. And taxonomy is the way we name all living things. All right, so I believe if they're underlined right there, that means you have a blank to fill in, yeah?
Yep.
So biological classification begins with taxonomy, the science dealing with the description, identification, naming, and classifying of organisms. Scientists are always naming things.
Yes. There's one named after Attenborough, David Attenborough.
Oh, that doesn't surprise me. Yeah, he's a pretty
It
influential.
was a pitcher plant, I believe it was in
Nice.
the highlands somewhere.
Nice.
Or like in the mountains.
This picture I have over here is just showing how we'll take something big and we'll break it down based on its characteristics into things that would put them into similar similarities between them. So it brings us to this guy, Carolus Linnaeus. Now Linnaeus was a brilliant guy, very interested in detail, and he wanted to give everything a name that everybody could understand. So he figured out a system of naming organisms. We call it binomial nomenclature. I think you have to write that one down.
Oh, wait, this is I don't have to write nomenclature.
Okay, just binomial?
Yep.
Okay. The naming of an organism with its genus and species name. So what happened here was that Linnaeus figured out that if he's an Englishman, but if he wanted everybody in the whole world to understand the way we name things, it would go back to the Latin language.
Yeah.
And so that's why he decided to use Latin, and I think it was pretty brilliant that he did that. He has something known as the Five Kingdoms system, which we're moving away from at this point. If you take biology in college someday, they'll probably work off of something called the Three Domain system, which maybe we'll talk about today. I think we are. But it still doesn't take away from the fact that Carolus Linnaeus did an incredible work by naming things. And it was a tough job. So let's watch a little bit of a video. It's kind of a silly video about it.
Before Carl Linnaeus came up with a way to name animals, they were given very long names that would describe what they looked like. An African ostrich, for example, might be called a large, feathered, long-necked, big-footed, backward knee-bending bird. This same long and useless name could also have been used for the South American rhea, and so we simply would not have known which of these big birds we were actually talking about. Linnaeus was into the business of naming living things so that we can recognize what they are. By being able to identify living things, we can work out how they are related to each other through a distant relation. For example, each and every one of us is given a first and a second name. Your first name identifies who you are, whilst your second family name identifies who you are related to. Since Linnaeus, many other scientists are still actively working today to name as many living things as possible, partly so we can understand how all life on Earth has changed over a long, long, long period of time. On Linnaeus' mission to name, he decided we should be given the most fitting one of them all, Homo sapiens, which means wise man. Homo sapiens is our first name and identifies us from all other living things as human beings. The name Hominidae is our family name. This name connects us to one of our closest living relatives, the orangutan, which also has the family name Hominidae. We are in the same family as these fuzzy orange tree climbers because we have many of the same characteristics, such as opposable thumbs that allow us to hold on to things. Two separate families of living things that look very different to each other can share a common ancestor to which they're both related. This is a bit like how you and your odd-looking cousin may look nothing like each other but will share grandparents on one side of your family. Therian mammal is the name given to a group of animals that are warm-blooded and give birth to live young, such as elephants, rodents, and whales. Another group of animals, called the reptiles, are all cold-blooded and lay eggs, such as snakes, lizards, and crocodiles. Both of these very different-looking groups, reptiles and therin mammals, share a common ancestor that lived over 300 million years ago, called a basal amniote. This animal was both warm-blooded like a mammal and laid eggs like a reptile. Linnaeus came up with a way to name animals as he believed that it was the responsibility of humans to discover, name, and understand all living things on Earth. But things aren't always easy, and naming can be a tricky business. Take the platypus, for example. They have webbed feet, and a bill like a duck, fur like a mole, and lay eggs like a reptile. Those poor, confused platypuses.
I think their claws are also like venomous.
The males. The male have venom in their claw, for sure.
They're
Yeah.
just the most confusing creature.
They are, and when the scientists saw that for the first time, they thought somebody had sewn things
Faked it,
together. They
yeah.
thought they faked it.
Because it's all the string.
And they're very private creatures.
Yeah,
They
so they're
come
hard
at, they're
to find.
nocturnal and they're very private, yes, so it's hard to
they're
find.
just very confusing.
I think we'll talk about them
Yeah, probably.
later in the year when we talk about mammals. They're in a very strange group called the monotremes, the monotremata. Okay, so the 5 kingdoms then is what set up, Linnaeus set that up by his women. His name was Carl, but Carolus, they call it, that's his Latinized version of Carl. So that's why he did that. Okay, so we have 5 kingdoms. Kingdom Animalia, which you know would include animals. Kingdom Plantae, which would be plants. Kingdom Fungi, because they were different enough from plants. Do we not have this in a spot too?
Do I put it in here? Like, I'm
What does
just
that
asking.
box say?
It just says classification system.
Let me see.
And the other one says memory codes. So I have no clue what that means.
I'm on the wrong page. Okay, do you know the other 2 kingdoms?
What? I don't know.
Okay,
I knew
you are not,
Plantae.
writing these in on those ones.
Okay. I knew Plantae and Fungi.
Okay, Animalia, Planta, Fungi. Then comes Protista, which includes protozoans and algae, and Monera, which they're changing to the eubacteria and the archaebacteria.
Okay.
So, but there's still, I still give a lot of credence to the 5 kingdom grouping. A lot of scientists want to change that. Some of them out of spite against Linnaeus because he was a Christian. And it doesn't mean that he was a bad scientist just because he was a Christian. So binomial nomenclature, of course, will bring it down to our genus species name. So for instance, this pebble crab would have the genus of Xanthias, the species Lamarckii. Yeah, so Xanthias lamarckii. And that's specific to this creature. Like when they said in the video, Homo sapiens is specific to human beings. All right, so here's what you're going to write down. Kingdom is the top one there. So today they would have the 3 domain system, but this is going to show us the kingdoms, phylum, class,
Okay.
order, family, genus, and species. So to the right you have a way to memorize it. Have you ever had one of those where you memorize these things?
No, not really.
Okay. So I've had a lot of students at OD I've had for years and years and years, and so we sometimes cover the same material, right? And so they all know that what I say is kings play chess on fine glass sets. See, that's a mnemonic that tells me kingdom and the P for phylum, C for class. And it, to this day, I always say, okay, kings play chess on fine glass sets. So I can figure out, what the next grouping is. So remember in the video it talked about a family name? That's literally talking about this family name right there.
Yeah.
Now, interestingly, this is the one that works for me that I've done for years and years. Kings play chess on fine glass sets. But in your book, they have King Philip's class ordered frozen grape sodas.
No, yeah, that just seems more convoluted.
I think this one rhymes a little bit. Kings play chess on fine glass sets. So if we go back to this one. So I don't know who came up with the grape soda one.
Yeah, no one really likes grape soda. It tastes like condensed purple.
And if it's frozen,
Yeah, especially that. That's the most confusing part. It tastes like purple is the best way to describe
it tastes like purple. Okay.
grape. Like, it does not taste like a grape.
So if I were you, I would go King's
Yeah,
Play
fine,
Chess
yeah,
on
yeah.
fine glass sets. Okay. All right. So, but it brings us to our classification system. Notice kingdom is second here because we have this thing called domain. That'll be talking about the 3 domain system, which is eukarya, archaea, and bacteria. So we'll get to that.
I didn't realize you were asking me. I was like, oh, hold on a second.
I'm not asking you, I'm just telling you. We'll get to that. But let's
Yeah,
take a
give
look
us
at this
pause.
critter right here. Let's look at all of his names he has. This is a red fox. And a red fox is in Kingdom Animalia, phylum Chordata, he's got a backbone, class Mammalia, he's a mammal, order Carnivora, eats meat, right? Family Canidae, which would be canine, right? Genus Vulpes, species Vulpes.
Yeah.
So when we call that his name would be Vulpes vulpes.
I can see it in the corner.
Yeah, Vulpes vulpes. And so I will tell you that whenever we write these down, so say you're writing down Vulpes vulpes, the genus is capitalized, Vulpes. The species is not, lowercase,
So
Vulpes.
it's just like an actual,
This is
just a
how,
start to a sentence.
and if you're writing it, you're going to underline it. You know why? Because it's in Latin. If you're typing it, you're going to italicize it. Is it italicized right there? Yes. See, it's italicized because it is, all of them, in fact, see right here, Vulpes vulpes, it's italicized because that is the Latin word, and that's how you distinguish if something's in Latin.
That is a weird way to do it.
I know.
It just seems odd to me to have to italicize it.
Yeah, and that's, but if you're writing it, you underline it. Okay? All right. Our 3 domain system is this, okay? So this is in your book on page 353,
Yeah,
and you're going to see that our 3 domains are these. Archaea, the second one is bacteria, and the third one is eukarya. And just personal opinion on I think this is ridiculous. That's because the vast majority of creatures are going to be in eukarya.
Yeah, but it's still like
Not numbers, but types.
Yeah, no,
Numbers, because bacteria are so
Yeah.
numerous.
And
Yeah.
it's still a good way. It's still just a way to divide things further, just to find them, I guess.
I know, I just think it's kind of, so then I take eukarya and I'm going to divide that into protista, fungi, animalia, and plantae. So there's my kingdoms, but I put them now into the kingdoms under the domain of eukarya.
Oh, hold on, I have to write this down.
Yeah, so this is your,
Well,
yeah.
Okay. Sorry, I just wasn't certain how to spell the first
And
one.
the reason there's only 4 kingdoms is because bacteria and archaea are in their own up here, and they would have been kingdom Monera in the old system. And I still call it kingdom Monera. Remember, I'm a microbiologist, so bacteria is my thing.
Don't you use Greek occasionally when naming things?
The Greeks certainly did, you know,
I mean, they're Greek.
I know, so I'm trying to think, what was the guy? Alexander the Great's teacher.
Oh, that guy, Plato, was it?
No.
Or, no, I know who you're talking about.
I keep trying to say Archimedes, but it's not Archimedes.
No. Was it that? No, it wasn't the guy who was on the
Aristotle.
street.
Aristotle.
Yeah, it was him.
Aristotle definitely named things. He categorized things before Linnaeus did.
Yeah.
And he put everything in Greek. Linnaeus said Latin would be more universal use. In fact, Latin is a dead language except in science, which is not spoken in science, unless you say Vulpes vulpes, right?
But you can still learn Latin. And I
Oh
suppose
yeah,
if you
yeah.
really
It's
try,
smart to learn it because we got a lot of languages that have
and
their
you can
names out of
break
that.
down scientific languages. I suppose if you wanted to, you could raise your child speaking Latin around them and make a native speaker.
Yeah, you could.
Instead of what happened with Hebrew, that's how it became a thing again. It
Oh,
became a
okay.
secondary
The
language.
Jewish
but
nation started
yeah,
speaking Hebrew.
it
so
was
don't-
whenever they got their
Smart.
language, they actually taught it to their kids and chose that as the main language.
All right, so in order to get to this classification of individual creatures out there, we go through something called a biological key. All right, a biological key can be a little bit
Yep, that's good.
tedious, but actually it can be kind of fun as well. Okay, so the page that you have in your book has a lot of them there.
Hold on, I'm just trying to.
We are not going to go through that. That's going to be homework for you. Okay, so what page is that on, Bradley?
68.
Okay, so it's going to tell you that you're going to use your biological key from pages 358, 359 in your textbook
Yeah, that's true.
to go through this. Now, I will tell you
That's fun.
that we're fixing to do one right now in class, but we're doing one that's not on your list. How about that? We're gonna do the elephant.
Okay, so where should I write that down?
Alright, so you might put it in conclusions here.
Okay.
But say, so like right here, we're gonna say, all the key is gonna do, it's gonna guide us through. Have you ever used a flow chart for anything?
I believe so, but I can't remember what it looks like.
Okay, so a
It
flow chart
sounds
is like a
right.
key. In other words, you answer a question. If it's yes, you go this way. If it's no, you go this
yeah,
way.
I think I've
Okay?
done it before.
Alright, so here when we have
Yeah,
vertebrate, we're
okay,
looking at
yeah, I've done
vertebrates.
that.
Do they have fur or no fur? So we're saying, well, the creature we're looking for has no fur. Let's say we're looking for this frog down here. No fur, so we go over here. Does it have feathers or no feathers? Well, no feathers, so we go down here. Does it have external fertilization or internal? External. No gills as adults, amphibian. It brings us to the toad. Okay? So let's watch one here, and it's about elephants.
Okay.
We're going to use the biological key to classify elephants. When we use the key, we always start with question 1, and our answer to question 1 guides us through the rest of the key. The first question asks whether the elephant is microscopic or macroscopic. Well, we don't need a microscope to see the elephant, so it's macroscopic, and that means we go on to question 3. Question 3 asks whether it's autotrophic or heterotrophic. Well, the elephant can't make its own food, it has to eat. So it's heterotrophic, and we move on to question 5. Question 5 asks whether it's a decomposer or a consumer. Well, the elephant doesn't decompose its food, it just eats it. So that means it's in kingdom Animalia, and we move on to question 6. Question 6 asks whether or not it has a backbone. Well, people can ride on an elephant, so its back has to be strong, so it has a backbone. That tells us it's part of phylum Chordata, and we move on to question 22. Question 22 asks about its mouth. The elephant has jaws because it can open and close its mouth, so that means we move on to question 23. Question 23 asks whether the skin is covered with scales or not. There aren't any scales on the elephant, so we move on to question 26. Question 26 asks what's on the skin. Well, if you look at the elephant on the right and look at its head, you can see hair. So there are feathers or hair, and that means we move on to question 28. Question 28 asks to make the distinction. Is it feathers or hair? It's hair, so we now know that the elephant is part of class Mammalia, and we move on to question 29. Question 29 asks if there are hooves or no hooves. We'll look at the picture on the left. The skin on the elephant goes all the way to the elephant's foot, so there are no hooves. That means we move on to question 31. 31 asks whether it's a carnivore or an herbivore. Elephants eat plants, so they're herbivores. That means we move on to question 33. Question 33 asks whether there are enlarged front teeth for gnawing. Think about a mouse. A mouse has enlarged front teeth for gnawing. An elephant doesn't have those kind of teeth, so we move on to question 35. 35 asks about the presence of a trunk. Well, the elephant definitely has a trunk, so it's in order Proboscidea. Notice there are no longer any numbers. That means we're done. We've classified the elephant as best we can with this key. The final classification then is Kingdom Animalia, Phylum Chordata, Class Mammalia, Order Proboscidea.
Okay, so you see how that works?
Yeah.
In your book, it's different from this one. This is from the old book, but it's similar. Okay, so you'll find it. So just remember, when you go through each one of those on your page there, you're going to answer the first question about it and then lead it down, and you're going to end up finding. So what it wants you to write then in the classification under, I think, let's say bison. I guess it's kind of close to an elephant. You're going to say Kingdom Animalia, phylum. What? It doesn't have that much room in there, but it says the number from
Yeah, it
the
really
key.
doesn't. That's why I'm like,
I'm not exactly sure what it means, number from the key.
Yeah, neither
Number,
am I. I don't
I guess
have
it would be number
I don't have
35.
Yeah, that's what I was guessing.
Yeah.
I don't have the big book with me, otherwise I'd pull it
Right,
out.
you could pull it out, but yeah, I don't have it either. So maybe
It's really
classification,
heavy.
it just wants you to do that last thing right there.
Maybe.
However
that's
far
what-
you can get.
That's what I'm guessing it's
Yeah,
saying.
and then you'll say number 25 or 35,
Yeah.
whatever it is. Okay, so yeah,
It's
do
really
that at
good.
home, and your conclusions are going to be something about classification
yeah,
and
and
how it
how
helps
it-
keep order,
yeah,
blah blah blah,
yeah.
something like that.
I took that out of my backpack because it's really heavy.
Oh, I hear you. I don't blame you at all for not bringing that book. Okay, so go ahead and turn the page, and we're going to answer some other questions now. Okay, because we're going to move into what used to be called Kingdom Monera. And now, I have this written wrong. When I say kingdoms, archaea, and bacteria, they're actually domain, archaea, and bacteria, not kingdom.
Okay.
All right, so they're called domain, archaea, and bacteria. All right, so let's see, we're going to watch a little introduction on these guys.
There has long been a delicate balance in nature. Without knowing it, we have lived in harmony with bacteria. Most of them are harmless and even beneficial to us, and play a crucial role in our world. These beneficial bacteria form an essential bond with us, helping us to digest our food, as well as to maintain our health by fighting off harmful bacteria such as pathogens. Yet we tend to associate all bacteria with disease and filth. The rapid increase in the world's population has been accompanied by a dramatic growth in consumption, leading to more food sources for bacteria. This means that bacteria are more likely to grow. When this happens with harmful bacteria, it results in excesses related to health, agricultural diseases, and overall hygiene. We have tried to solve these problems by killing the harmful bacteria using antibiotics, pesticides, and chemicals. But every time we use these measures, we kill almost all bacteria, both the harmful and the beneficial ones. Only the strongest survive, and they have more space to grow due to the lower microbial diversity. New strains evolved that are more resistant to the higher chemical doses that we apply. As a result, strains have even evolved to a level that they are completely resistant, unaffected by even the highest dose, and it becomes superabundant due to little competition from other bacteria. This has serious implications for our health and our environment. We are breaking our ancient pact with bacteria, with major unpredictable consequences. We need to understand that what bacteria require to grow is not affected by the use of antibiotics, pesticides, and chemicals. It requires a new approach. We must restore our alliance with the bacteria and exploit their beneficial qualities. A group of bacteria with many beneficial qualities can be found all around us, members of the Bacillus family. Scientists at Noble Bio have developed a system that makes it possible to find, isolate, screen, and grow different types of bacillus. Individual strains of bacillus species are grown in fermentation tanks and made into spore form so we can easily reintroduce them into the environment, where they can restore the microbial balance. Because of their diversity and strong characteristics, they are highly suitable for a wide range of applications. They can help to improve hygiene by degrading the bacterial food source, for example, thus limiting the need to use aggressive chemicals. In agriculture, they can help to improve the health and digestion of livestock. When beneficial bacteria populate the intestines, they can prevent harmful bacteria from binding, a process known as competitive exclusion. Thus, diseases can be forestalled and livestock grows more efficiently. They can help crops grow by making it easier for the roots to take up nutrients and by preventing diseases. This enables us to reduce the use of chemical fertilizers and pesticides and to produce healthier food. Bacillus bacteria are powerful and can be used to sustainably combat a wide range of the world's problems. By fundamentally changing our view of bacteria, making use of their qualities instead of fighting them, we are able to restore the microbial balance. A balance that affects all life on Earth and is of profound importance for future generations. Noble Bio.
Okay, anyway, so that ended up being an ad there at the end. It almost sounded like one of those movies, you know, where that computer person's talking to you. It's a little bit dystopian or whatever.
Yeah.
Now, the reason I have that in there is because I have a huge respect for bacteria. I love bacteria. I'm a microbiologist. I like it. And they get a bad rap because there are those few that cause disease, right? We call those pathogens. Next week, we'll talk about some of the famous pathogens, okay?
Yeah,
because they're
Plague.
Real. Like what?
The Black Plague.
Absolutely.
The
Yersinia
one
pestis.
spread through mice
Yes,
and
we'll
whatnot.
talk about that next time.
No, it's interesting.
but-
is, the fact that they were trying, like, they thought it spread through air or something like that, but
Oh, sure.
they thought
Miasma,
mice filled with
yeah.
air. So they were right for certain things, but they got it really wrong on why.
True, true. So it took some
Which is
time to
just
figure
really
that thing
funny
out,
to
and
me.
then we will cover some of that next week, which is some interesting stuff to me. But we have to remember that most bacteria are either
Or
harmless
neutral,
or beneficial for you.
yeah.
For instance, there's bacteria all over your skin right now.
Yeah.
And you think that's gross. Well, no, it's not. It's keeping fungi from growing on your skin. Okay, it's keeping your skin healthy. So it's good for you. In our gut, we have all sorts of bacteria. If we didn't have that, we couldn't digest our food.
Yeah.
We couldn't
It's
make
actually-
certain vitamins that are made in our bodies.
It's actually something that some people have where they don't have enough bacteria
Sure,
in their gut,
yeah.
so they actually have to have a doctor insert some.
Absolutely,
It's
absolutely. In
just,
fact, probiotics are part of that. I take probiotics every day, and that's actually swallowing a capsule that has bacteria in it. Yeah. So if you eat yogurt, same kind of deal. Okay, so when we talk about prokaryotes, remember there's two different kinds of cells, eukaryotes and prokaryotes. In archaea and in bacteria, we have the prokaryotes. Okay, so archaea and bacteria are prokaryotic cells. And what that means is they do not have a nucleus. In fact, sometimes when students ask me how to define a prokaryote and you say it doesn't have a nucleus, that's enough for me. I'll accept that. It has no membrane-bound organelles. We made the big cookie cell model, right?
Yep.
And remember we had all those things like mitochondria and ribosomes and nucleus
One
and
heck of a word.
all those crazy things. Well, they don't have that in the prokaryotes. They don't have those membrane-bound
Yeah.
organelles. But an easy way to say it is they don't have a nucleus. So what that means is that DNA or RNA, mostly DNA, is just floating around freely inside the cell. They do have ribosomes, notice. Do you remember what ribosomes do?
Give me a second. It's like a code for something, right?
Did you say a code?
Like a code to produce
Yeah,
something,
they
right?
take the code and make a protein.
Okay,
Remember,
so
they make
something
a
that
protein
takes-
synthesis in the ribosomes.
Sorry. I
The
couldn't
code
remember
comes
what it
from the
was.
DNA.
Okay.
It gets translated by the ribosomes into a protein. And we did that little deal where you had to make a sentence, remember?
Yeah.
That was protein synthesis. We had to go out of the nucleus into the ribosome. Yeah. Also notice that some of the prokaryotes, some of the bacteria have capsules. I will tell you that the ones, some of the most famous pathogens have capsules. And the capsules make them sticky. And they stick like, say, the back of your throat. And
Yeah.
that could give you strep throat or something like that.
Yeah.
We're going to look specifically at this cool thing. The flagellum is like a little tail right there. They do have cell walls. Some eukaryotes will have that, namely plants will have a cell wall as well. Everything has a cell membrane. All eukaryotes, all prokaryotes have a cell membrane that helps delimit the cell. It helps to tell you the shape it's going to be. And then it floats around in something called cytoplasm, just like with our regular eukaryotic cell. The archaebacteria, another name for these, are called extremophiles. So the blank you're filling in is the
Okay.
word extremophiles.
I was just
That
trying.
means they love extreme places. They live in harsh/extreme environs, which is a fancy word for environments.
Right?
Yeah.
I'm just gonna pull up harsh and.
Yeah,
not-
you can just say
not
harsh.
put harsh and extreme because it's just really long.
Okay, so this is why I'm a little confused as to why they've changed the nomenclature to the idea of the three domains. Because the archaea are very unusual bacteria. They need to be in their own group for sure, but
Yes,
they're
well,
still
they do.
prokaryotes. But there's not that many of them. So I find it unusual that we're having this whole domain named after this not very commonly found stuff.
Just remember, there's such thing as this like huge single cell organisms that you can see with the naked eye. It's like
Huge
these
what?
Single cell organisms. It's like a single
Like
cell,
an ostrich egg.
and it's just giant.
Yeah.
Yeah.
Depends on what it is, yeah. Not these guys. They're small.
Yeah. No, I was just remembering that.
Okay,
Like
can you see what's happening in this picture? This is
Um,
like water,
yes, I
right?
believe it is algae, right? Growing.
Actually, that's the archaebacteria. Okay? And they have this.
Some other things that I've seen where algae
Sure,
grow in them.
right. So what's happening in this particular spot is very harsh, extreme circumstance. What we have here is heat and a bunch of sulfur.
Salt. Oh, salt. Yeah.
And so
Can't put
normal
everything.
things don't like to grow in that.
Yeah.
In fact, sulfur is a group of drugs that are antibiotics, right? But the archaebacteria can handle it. They can grow in this. All right, so the three main types we're going to be looking at, this is in your next boxes down there, are ones called methanogens. They live in swamps where there's not much water. They create something called swamp gas. You can guess the name of the gas. It's called methane. That's what they're called, methanogens. And then we have our halophiles. Halophile means they like salt. So the Dead Sea is too salty for things to grow, but the Great Salt Lake in America is a little less saline, a little less salinity, and so some stuff can grow there. And what can grow there? Some halophiles, some bacteria, archaebacteria. Normally salt kills living things.
Mm-hmm.
In this case, they can grow in that salty environment.
Yes.
See, it says Utah Salt Lake, the Great Salt Lake. And then we have the thermophiles, high temperature. Okay, most things don't like to be too hot.
No.
Think about the caldera of a volcano. That's hot.
Yeah.
We have some of these bacteria growing there in the caldera of a volcano. I'm not kidding you.
Or the what's this, like Death Desert is something like that. Or,
Yeah. Well, now, okay, I will tell you, all bacteria, whether they're archae or regular eubacteria, eu means true, is the biggest enemy of all of them is desiccation or drying out. So they don't want to be in a dry place. They want to be in a nice, moist, kind of dark place. But in this particular case, like I would say, probably archaebacteria can't live on the surface of the sun. I don't think anything can withstand that, right?
No.
But that on the Earth, those very, very harsh environments, the archaebacteria say, no problem, I'm going to go live there. Okay, so now you got those boxes filled in with all three, yeah?
Yes.
Okay.
Unless you want to be right when they're located.
No, you don't have to do all that. We're just talking about where we found them, okay?
Okay. Wanted to make sure.
You understand that thermophile means high temp, though?
Yeah.
Okay.
Because thermo means like heat or something like
Good.
that. Or
Right, and
temperature.
halo, high salt, because halo
Yeah.
means salt.
Yeah.
Okay.
Or something
So
like that.
bacteria, now switching over to bacteria, also known as eubacteria, EU bacteria, EU meaning true. These will be the vast majority of the bacteria, the ones you've heard of, strep, staph, all those are in this group. Bacterium is the singular for bacteria. Bacteria is plural. And that's because there's tons of them. Whenever you see bacteria, there's not just one single bacterium. They're growing in a big group.
I'm just trying to figure out what prokaryote is, sorry.
Okay, bacteria are organisms made up of prokaryotic cells. Oh, you don't have that blank to fill
I
in?
don't think so. Are
Okay,
all
I think I
bacteria-
just repeated it, Riley. I think I just repeated it
Okay.
from earlier deal. A pathogen, though, is an organism that causes disease. Sometimes bacteria can be that. Okay, they can be a pathogenic. But there are other things that cause disease too. Fungi cause disease. Viruses cause disease. They're not living, of course. So parasites, exactly. So, but some bacteria.
Are really touchy because they have most of the things to make them alive. They just can't reproduce by themselves.
There's a couple of things they can't do.
Yeah, there's a couple of things they can't do, but they're pretty dang close to being alive.
They're
so
close,
it's,
but
it's really weird.
they have to kill a host. Well, not kill a host. They have to use a host.
They just have to use them, yeah.
So here's my question: are all bacteria pathogens?
No.
The answer is a big
It's-
N-O.
No.
No. In fact, most are beneficial. This is
Oh, I
me.
just look good.
This is me taking it up for the bacteria out there, right? So that's my microbiology heart right there. So I like bacteria. Okay, you're going to draw this next thing right here, a
Oh.
typical bacterium. And I will tell you, it is a rod-shaped bacterium, which is also known as a bacilli or bacillus. And so, like what they were talking about in the video, it looks like this. So you're going to basically draw a hot tamale.
Sorry, I'm just trying to
Or,
figure out what
you know,
way to draw it.
We call it a rod, a capsule, like a capsule. All right, and you're going to see the DNA is floating around in there. So what do I want you to include on there? I want you to include DNA. Put a little tail on it, called a flagellum. You don't have to necessarily put. You can write capsule for this outer part right here. These little things sticking off called pili. Pili are another way of making them sticky. They can stick to whatever they're infecting, if you will, along with that capsule.
Accidentally went out of the box.
So, but we're going to look at, even though we call this a typical bacterium, we're gonna look at they come in different shapes,
Just have to.
but all of
kinda
them are
seal it up a little
microscopic.
bit.
Yeah, all of them need a microscope to see.
Just trying to figure out what ways to draw the things. It's a bit tough. I mean,
Yeah,
it's-
that's the thing about biology, it does require some artistic.
And pal, I have none of that. Like I really tried a lot. I just can't.
Yeah, but in biology class you still have to go for it. So
To try and do that. Yeah.
so I think make sure and put your tail on there called the flagellum.
Yeah, I put the little tail on there.
Yeah.
Not very well, but I just sort of.
We'll talk about it in some more detail here in a minute.
Yeah. Do I have to make the several walls?
No,
thing, or-
no, just put cell membrane or
Okay, hold on. I gotta.
we just call it they call it here a plasma membrane. Notice they called the stuff in there called cytosol, but we can call that
Flagell. I'm just gonna label the
Cytoplasm.
flagellum as just flagell because I don't have enough space.
all right.
Uh,
You might have to write that word here in a minute anyway. So
okay. Give me a second. Spelled it wrong.
I don't count off for spelling. So.
Oh, I just went garn or gain instead of.
Okay, we're gonna move on. If we have to come back to this, I'll do it, and then I can turn the thing off. Okay.
Hold on, I'm just trying to figure out one last piece. Okay, chromosome. Okay.
You could just call that DNA. Just call it
There's
DNA.
another part that's called DNA, the plasmid.
But that's a plasmid. Not all of them have
Oh,
that.
okay.
They don't all have that. But if you drew it in there, that's fine. Okay. So the bacteria shapes, however, they can come in different shapes. We have spheres known as cocci. You've heard of staph is staphylococci or staphylococcus. Streptococcus.
I'm trying to find the area.
We have rods, which are called bacilli, and we have spirals. So I think in your book, you're going to write
This is what I thought.
sphere, rod, and spiral. As long as you remember, sphere is coccus, rod is bacillus, spiral is normally called spirillum, sometimes called spirochetes.
Oh, it's sphere. Sorry.
Sphere.
I heard you say sphere.
Well,
sorry.
these are spirochetes down here, which are spirals. So you can see like these some of these rods have flagellum on. This is a flagellated rod. Here's a spore forming rod. Ooh, Clostridium botulinum. We'll talk about it next time. Causes botulism. One of the deadliest things known.
Botulism. Sorry, that just sounds
Whew,
like a made up
yeah.
word. It
No,
sounds like
it's
a-
real.
No, botulism. Just that sounds like you botched something.
Ah. All right. So the three things: sphere, rods, and spiral.
Spirals,
You got
yeah,
them?
or-
Okay.
Yeah, spirals.
All right, so how do we classify bacteria? They're classified by shape and cell wall structure. And it turns out, because of their cell wall is very distinctive, some of them will take on a staining procedure that others won't. So we call that a Gram stain, named after Mr. Graham, G-R-A-M, Graham. Next time we get together, we're going to look a little more closely at the Gram stain. We're going to probably stain something, and I'm going to send home with you something to get a culture from home somewhere, and you'll bring it back the next week. All right, so if they're Gram positive, they hold on to the purple stain.
Yeah.
If they're Gram negative, they don't hold on to the purple stain, and they end up looking kind of with a counter stain called, it's red. All right, so cell wall structure is tested with a Gram stain. Do you have these blanks?
Yep.
Okay. Those that appear purple after staining are called Gram positive. Those that appear red or pink after staining are called Gram negative. Antibiotics are prescribed based on the type of cell wall pathogenic bacteria have. In other words, are they Gram positive or Gram negative? We can test that reasonably quickly, and then we can see what antibiotic would be best for that. And this is just a hint right here for the alliteration. Purple means positive.
Purple's positive.
Yeah. Red's negative.
Alright.
So think
I
about
can
negative,
probably-
red being
I can probably just remember red negative and.
Yeah. P positive,
Yeah.
purple positive. Okay. So here's our positive is purple, it's all happy, it's holding on to that purple stain. Gram is negative, it's sad because it couldn't hold on to that purple stain.
It looks more upset.
We're gonna watch a little video on, this is a goofy video, forgive me, but it's informative.
Bacteria have cell walls made up of polysaccharides that give them strength and rigidity. This is important since bacteria often experience variations in osmotic pressure due to the different solutions they encounter, and it is their cell walls which prevent them from shrinking or swelling. As a reminder, osmosis is the process by which solvent molecules pass through a semi-permeable membrane from a less concentrated solution to a more concentrated one, equalizing the concentration on either side of the membrane. Nearly all bacterial cell walls have a peptide polysaccharide layer called peptidoglycan, also known as murine. Peptidoglycan is a polymer made up of sugars and amino acids which forms a kind of mesh. Bacteria can be classified based on their reaction to the Gram stain, which identifies them as gram-positive or gram-negative, based on the chemical and physical properties of their cell walls. Gram-positive bacteria have a thick cell wall, which consists of up to around 30 layers of peptidoglycan. This cell wall surrounds a monoderm, which is a single plasma membrane. Gram-negative bacteria have a much thinner cell wall, consisting of a single layer of peptidoglycan. This layer of peptidoglycan is sandwiched between two lipid bilayer membranes called diderms. We can differentiate between gram-positive and gram-negative bacteria by dyeing them with crystal violet and then washing them with a decolorizing solution. Then a counterstain is added, for example, safranin or fuchsine. Gram-positive bacteria will retain the crystal violet dye and remain purple, while the gram-negative bacteria will be stained pink. Note that gram-positive bacteria also pick up the pink color of the counterstain. However, this is not visible when they are dyed with the darker purple color of the crystal violet stain. The reason for these staining differences is due to differences in cell wall structure, which is the chief difference between gram-positive and gram-negative bacteria. The gram stain detects peptidoglycan, and since gram-positive bacteria have a thick, multi-layered peptidoglycan layer, they retain the crystal violet dye. Gram-negative bacteria do not retain the dye for two reasons. They have an outer membrane getting in the way of the crystal violet, and they lack peptidoglycan to retain the stain. Although both gram-positive and gram-negative bacteria can be pathogenic, gram-negative bacteria are more resistant to antibodies because of their impenetrable cell wall. Unfortunately, these bacteria also develop resistance more quickly. Not all bacteria can be reliably classified through gram staining. Acid-fast bacteria and gram-variable bacteria, for example, do not respond to gram staining. Acid-fast bacteria are bacteria whose cell walls retain stains particularly well. Although they aren't closely related to gram-positive bacteria, they can appear purple after the gram stain test. Gram-variable bacteria show a mix of pink and purple cells when stained.
Okay, that wasn't as goofy as I remembered it being. Okay, so sometimes also your technique doing the gram stain can make them end up looking sort of purplish pink. So it can be a little bit iffy there. All right, but notice here it's pretty purple right there. And then we have our pink or reddish ones right over there, okay? All right, movement. The flagellum is an engineering masterpiece. That's what you're writing there, masterpiece. And it is very complex. All right, so bacterial movement is based, they can have no movement, they have a flagellum, or they have what are known as slimy threads, which, but they do move around. They can move around. For the flagellum to come about accidentally, evolution would require dozens to hundreds of precisely tailored parts made by protein, all made by accident at exactly the right time. Otherwise, the unused parts are waste of precious resources. So really, the honest truth is that this is what it looks like. This is a microscopic bacterium, and it's got this machine on it that drives this little tail to spin around. Look at all those different things that have to be working just right. If the bacteria doesn't have it, it won't survive. Now, there are a couple of ways of working it. One of them, it causes it to spiral around. Like a spiral like this can just cause itself to move around. That's not as advanced as the other one. The other one looks something like this. Look at the machinery there. To turn that, and it turns it kind of like a corkscrew. And it makes that flagellum move around like that and gives it motility. And motility, you would agree, would give it some advantage for survival, right? Yeah, so it's a pretty amazing thing right there. There have been people who have studied the mechanism to this, and become believers in God because of it. Because there's no way it could have happened accidentally. All right, cyanobacteria. That's the next big word you're writing. Cyanobacteria is essential for oxygen production on Earth. It's usually called blue-green algae, but the truth is it is not algae, it is a bacterium. It's a eubacteria. It is very common. It produces over 85% of the oxygen we breathe. So it's not, blue-green algae are not actually bacteria. In fact, I have a slide for you to look at, blue-green algae. Today, they're almost always called cyanobacteria, which is a better description. They are photosynthetic. They're producers, providing much of our atmospheric O2. And they come in different groups like this. We can see these different ones here. Ones that look more round, ones that look more like strands or like spirals. Pretty cool.
And then a square.
And of course, remember photosynthesis? You remember this? Yeah?
No.
6 CO2 plus 6 H2O gives me C6H12O6 plus 6 O2.
I see it. It's just that it throws me off. I'm
Really?
trying to remember it. To
Okay,
remember it.
so you might not be chemistry minded. If that
No,
confuses you.
I'm not
All right.
super interested in chemistry.
So it's time for lab, and what we're going to do for lab today is to look at a few scopes and microscope slides, and you're going to draw those in your book, okay?
Yep,
Next time we get together, we'll do some talking about how we culture bacteria and things like kind of right up my alley.
Science.